Okay, hello everybody. If the President's ASLO permit, I would like to change the meaning of ASLO for 1 hour. Okay, I want to change the meaning of the ASLO from original meaning to amazing scientist in limnology and oceanography. Okay, let me introduce the amazing scientists in the field of oceanography, especially biological oceanography. Professor Julie LaRoche from Leibniz Institute of Marine Science, Germany. Let me introduce her brief experience. She got PhD from Dalhousie University, Canada in 1986. She has been awarded a Canada Research Chair Tier 1 in Marine Biogeochemistry and Microbial Genomics. She has produced a lot of excellent research on nitrogen fixation in the ocean, phytoplankton dynamics, role of microbial communities on biogeochemical cycles. The title of her talk today is Future Aspects of Research on Marine Nitrogen Fixation. Please welcome.
First of all, I'd like to thank the organizer for giving me the opportunity to come here in this beautiful country and beautiful venue to present this plenary talk. And I hope I will get other opportunities to come back and visit your country in more depth. It's very interesting so far. As was mentioned already in the introduction, I'm a marine biogeochemist, and I use molecular techniques to look at various aspects of marine biogeochemistry. Today I'm going to talk about marine nitrogen fixation, and so various aspects of it. How do I change the slides? Sorry. Like this? No. Help. Oh, there we go. This is the various topics I'm going to cover during my talk. Why marine nitrogen fixation is important, what we know about the diversity of the isotopes, what we know and maybe don't know about the distribution and activity of the isotopes on a global scale, and how. And I want to finish my talk with giving you some very recent results that show that the 1592 tracer method that was used widely to measure nitrogen fixation rates in the ocean has been probably underestimating the true nitrogen fixation rates. Then finish with some pressing questions and how we could address them. So from a marine biochemistry point of view, the nitrogen cycle is very important because it has some major control on primary productivity. So the marine fixed nitrogen inventory of the ocean is controlled mainly by microbial processes of loss and gain processes. Dinitrogen fixation or nitrogen fixation is a gain process, and it takes them to gas, and then the nitrogenase enzyme converts it to ammonia, molecules of ammonia, and with a lot of ATPs and then electrons are needed for this reaction. In contrast, we have also some loss processes, anamox, or anaerobic ammonium oxidation and denitrification, that take the fixed nitrogen and release it as into gas. So why is this important? The fixed nitrogen inventory then is controlled by these gain and loss processes, and there's been ongoing debates in the literature and among geochemists and biological oceanographers as to whether the marine fixed nitrogen inventory and then controlled by these gain and loss processes is in balance and remain constant with time. So the geochemists are arguing on the basis of isotope studies that the nitrogen inventory is fixed with time. And if we look at the rates, then it looks like the nitrogen gain are much lower, what we're measured is much lower than the nitrogen loss gain. So that means that over time, we'd be losing some nitrogen from the ocean. This is not responding. Am I doing something wrong here? Ah, there we go. So then that the fixed nitrogen inventory would be reducing its size over time. So this is in one aspect of why we need to the nitrogen inventory is very important, and the fixed nitrogen inventory is very important in controlling the primary productivity in the ocean. You can see this from the surface. This is a map of the nitrate concentration in the surface of the ocean, and large regions are depleted in nitrate or fixed nitrogen in general, and therefore nitrogen is a limiting nutrient in the ocean, controlling primary productivity. Thank you. Okay. So we need to know something about the diversity of the diazotrophs, and by far the best characterized diazotroph is trichodesmium. And this is what you can see here is a bloom of trichodesmium. And this is a filamentous cyanobacteria that fixes nitrogen. We have a lot of very excellent work that's been done on this to understand the cellular biology, the physiology, and also the distribution, the factors controlling the distribution of trichodesmium in the ocean. It's isolated in pure culture, and we have also genome available for this. Trichodesmium is found in the open ocean and also in the coastal areas. You can see here along the Great Barrier Reef here, there's a bloom of trichodesmium there. So this is what it looks like under the microscope. So it looks like tangled hair, but it's really filaments here. And this is associated with atmospheric dust particles, sarin dust particle here that provide iron for the trichodesmium. So seeing is believing, and then so it was relatively easy to identify the first diazotrophs because we could see them under the microscope, and people who worked on these could see them in the microscope. So we know that the trichodesmium, the non-heterocystis trichodesmium, is a nitrogen fixer, and also in the large plankton we have associations of some heterocystis Cyanobacteria Richelia with diatoms. You can see here the heterocysts and the little filaments. And so those were early on recognized as important diazotrophs in the ocean. But what we didn't know is that there was a whole suite of other diazotrophs that we couldn't see because they're just non-descriptive. They just look like all the other cells that are all the other bacteria that are in the environment. And it's not until we were able to use molecular techniques and to amplify the NIF genes, which encodes the nitrogenase enzyme, that we were able to experience really understand the diversity, the broad diversity of the marine diazotrophs. So you can see here the nitrogenase enzyme, and this is the gene that the part of the protein that we use to design primers to amplify the nitrogenase, the NIF gene in the environment. Doing this, we found more cyanobacteria that are diazotrophs. So here you see Crocosphaera, which is a unicellular cyanobacteria. And also we found some very abundant other cyanobacteria. They're very small, and then these are not in culture yet, so we don't know what they look like. They're best identified as a flow cytometry chart, like right here. This is where you can concentrate these organisms. Very recently, there's been incredible progress in the sequencing techniques, and then we can use high throughput techniques using tag sequencing and then sequencing NIF amplicons very deeply. And then this is where, at this time, that we've really realized the true depths of the diversity of the diazotrophs. And you can see here we have several clusters of diazotrophs. Here, in the green, you have the cyanobacterial cluster, but you can see that by far the most diverse cluster is the protobacteria. And for this, we don't have any cultured representatives of diazotrophs that we can based our research on. So, how many of these organisms are important in the ocean? Well, if you want to address this question, you need to be able to count the cells in some way or another. So, again, if we go back to trichodesmium can be kind of difficult, but it can be counted under the microscope. And we have some fairly good understanding of the distribution. At least we know where to find it. This is where most of the research has been done in the past. So this is a compilation that a grad student and I did in 2006, and there's a much more detailed one that came out recently by Luo and Scardoni and his group. And that included most of the researchers working on nitrogen fixation. But you can see here, this is a 25-degree isobath here. And then you can see that most of the trichodesmium falls within this 25-degree temperature range. It's not so easy for the rest of the diazotrophs, especially since we don't know what most of them look like. But also they're not very abundant in the microbial community. So if you look at the most abundant microorganism, phytoplankton in the ocean, which is Prochlorococcus marinus, is found in an abundance of 10 to the 9 cells per liter. In comparison, on this page here, you would find there would be 1 cell of Crocosphera watsoni, which is one of the unicellular diazotroph, at about less than 10 to 6 cells per liter. So assuming there's about 1,000 cells here of Prochlorococcus. So if you want to use microscopic techniques to try to identify the diazotrophs, it's going to be very difficult. They're very rare. It's like looking for a needle in a haystack. So you have to basically pull the diazotrophs out of this microbial community, and then the easiest way to do this right now is with molecular techniques. And we use something that's related to PCR, which is called qPCR, quantitative PCR, that allows us to, by knowing the diversity of the NIF genes, we can design primers and then probes that will target specific groups of the diazotrophs. So a number of people have done this, including my lab. Oh yeah, by the way, this is like a little thought bubble here. How does NIF copy numbers relate to cell number anyway? We don't know that, but at the moment we don't have any other way to address this problem. So until this is something we need to resolve, but until then, if we want to see what the diazotrophs are, I think qPCR is one way to go. So, as like I said, a lot of people have done this, including my lab, and I just want to show you a few examples of this type of result and what we can tell from it. So once you start looking at the distribution of the diazotrophs, you can also tell what their environmental preferences are. So if you look at the panels here, so all of these panels are expressed in NIF copy numbers per liter on a log scale. So 10 to the 6 here will represent a million cells per liter. So the total, million NIF copy genes per liter. So the total NIF here would be all of the different groups that we've targeted. So you can see this is in the eastern tropical North Atlantic, and you can see it's pretty much covered at 10 to the 6 per liter throughout. If you look at the separate groups now, if you look at trichodesmium probes or UCNA, which is the mystery organism, Procosphera, which is the unicellular cyanobacteria, or one of the proteobacteria, you can see this one is also widely distributed throughout the North Atlantic. But one thing that comes out is that you can see that, for example, trichodesmium follows a very strict distribution according to the temperature. Here we have temperature, and then this is about 27 degrees centigrade here. You can see this is a cut where we get an increase in trichodesmium. And then a while back, with some grad students at Bradbat, we did some growth curves, growth experiments that showed that trichodesmium has a growth optimum at 27 degrees centigrade. So this confirms the distribution that we observe in the field as well. If you look on the depth profile of this, here we can see again the segregation between trichodesmium and the UCNA organism here. What we have shown here, instead of showing the NIF copy numbers in the DNA, we show it in the RNA. And then looking at the RNA gives you also an indication of the expression of the gene, so the activity of the bacteria, although this is not really convertible to a rate of nitrogen fixation. Right, so like I said before, there's been a very detailed compilation of the data on marine nitrogen fixation, and this is by Luo Doni, and this is on a discussion paper on the web. And you can see here what they've showed the spatial distribution of the data, not the measurements, but just the distribution of the data on marine nitrogen fixation. And I put this slide here to show you that we have been very biased in sampling the ocean for the diazotrophs. And what you can see here, these big spots here, that's probably bats, and that's probably hot station or the time series station. And then most of the data is in the western tropical Atlantic, which reflects a little bit the distribution of the scientists who worked on nitrogen fixation over the years. When I started working on nitrogen fixation in Europe, I proposed to work in the eastern tropical Atlantic, and everybody told me, well, don't go there, there's no nitrogen fixation there. But of course, this was because there was no data there. But once you start collecting the data, you can see that this also was really happening there as well. So, the bottom line of this slide is that there's large areas of the oceans that are not covered by the currently available measurements, and then we need large-scale measurements to understand the large-scale processes in the ocean, and we also need a dimension of time, and there's some stations that are established here to address this. So I want to give you a couple of examples from regions that have been undersampled, and then what we can learn from looking at the distribution of the organisms there. So and one of the areas is the South Atlantic here, and then there's very, very little data there. We've been going on these long cruises where we can collect along the way a lot of data points, so to look at the distribution of by qPCR, and we found that there's, if you look at this distribution here, this is for the UCNE group, which is the unknown, the uncharacterized, unisolated cyanobacteria. You can see in the South Atlantic here and in the North Atlantic, you can see that at a certain time of the years in the South Atlantic, there's huge blooms of these organisms. So we have 2 times 10 to the 6 copies here of the NIF gene per liter, and it's also really highly expressed. Another point about here we have the well-known trichidesium distribution. You can see that trichidesium clusters in the regions of high temperature, above 26 degrees again. So this follows well the pattern. So between the 2 cruises, we could see this is the temperature line here for one of the April cruise that we went, and then the other one is for the October cruise. You can see at each time trichidesium follows is above a temperature of 26 degrees. And then the UCNE distributions are on the outside of this range, so there's separation between these 2 groups. What we measured for nitrogen fixation rate at this point here was 12 nanomolar of nitrogen per day, and that is quite high for nitrogen fixation rates. Another part of the ocean which has been undersampled is the South Pacific, and I want to show you some data from there, because this is interesting this time, because we now know there's a lot of non-cyanobacterial diazotrophs there in this region, in the oxygen minimum zone of Peru. And by doing some phylogenetic studies, we've identified 8 new groups of non-cyanobacterial diazotrophs that we also characterize by qPCR to quantify them. Some of the groups have no close relative, and then some are among the protobacteria. Again, if we look at the distribution of these various groups here, so here we have one we call P1, one we call P8. You see a little bit of separation again in these distribution. This becomes more pronounced if you look with depth profiles. So let me locate you a little bit here. This is the transect we're looking at, and this goes through the oxygen minimum zone of the Peru upwelling. And you can see here we have the nitrate concentration, so high nitrate concentration at depth here, and then oxygen are very low here below very shallow, about 50 meters. The distribution of one group of these organisms is right in the very, very high nitrate concentration. And then this again goes against the general thinking about the nitrogen fixation, that nitrogen fixation should only occur when you have no fixed nitrogen available. We did some experiments there in the Peru upwelling, adding glucose and then various manipulation experiments, and we showed that these groups of non-cyanobacterial diazotropes can be stimulated by the addition of glucose. Now, I just want to finish my talk, and that's good. I still have 6 minutes. I think I can make it. By presenting some data on the nitrogen fixation measurements, and then why we think they've been underestimating the true nitrogen fixation rates over the last 15 years. So, there are two accepted methods to measure nitrogen fixation in the ocean. There's, nitrogen fixation in general. There's acetylene reduction, which uses acetylene, which is an alternate substrate of the nitrogenase enzyme, and then acetylene is produced, and then this is measured by a gas chromatography. Or we can add 15N2 tracer gas and follow the incorporation of the 15N2 tracer into the particulate organic material. Turns out this is again from some data compiled by LaRoche, Donny et al. And then it turns out that most of the nitrogen fixation rates in the ocean have been measured by the 15N2 assimilation method. In 2010, we published a paper that showed that says that the 15N2 tracer addition method would have led to some large and variable underestimation of the nitrogen fixation rate as it's applied currently. And I have to say from the start that I can't take any of the credit for this discovery, whether that's good or bad, but my PhD student, Viv Kumar, did most of this work. I was on vacation when she discovered this, so I kept getting this frantic message saying, there's something wrong with the method, I can't balance my growth rates in my experiments. So this is how it happens. This is what you do when you measure nitrogen fixation with the 15N2 method. You have a water sample which has a natural abundance of a 15N of 0.366%. You add 99% 15N gas to the water sample and as a gas bubble, and then in theory, you should reach an enrichment level that should be about 5 to 10 atom%. And then this is incubated with the microbial community, and then after a while you filter and then follow the incorporation into the particulate organic nitrogen. So the experiments that Wiebke was trying to do was to follow the incorporation of 15N2 and Crocosphere in the lab. Normally in my lab we use acetylene reduction for lab experiments because it's faster, we get the results faster, but this time she wanted to use this for a particular reason. And she was finding that the rates of nitrogen fixation were about 1/10 of the growth rate that she was measuring, so there was something wrong. So when I was on vacation and not really thinking very much, I said, well, it's a factor of 10, it must be an error in your calculations. Go back and redo the calculations. So, but of course, when I came back a week later after my vacation, she had figured everything out. And then she came in my office very confident and said, well, there's only one thing I don't measure in this whole equation to calculate the nitrogen fixation rates, and it's the enrichment in the seawater. This is a term that's calculated from a theoretical calculation from the amount of gas that's added. And so then she figured out this assumption was probably not correct. Then we did some experiments just looking at the equilibration time of the isotope in the seawater, and found out that it's very slow. And then it takes up to 12 hours, but doesn't even reach the 100% of what you expect even after 24 hours, so that takes several days. So at the time we had many grad students doing a PhD looking at nitrogen fixation in the ocean, and then so what do? we have crews going on, and then you can't send a grad student to do his PhD with a method that doesn't work. So we stopped basically everything and tried to come up with a better method, or a method that we thought that would give us a constant enrichment in the seawater over time. And so quickly, well, with a lot of work, we designed a method by degassing water, adding the gas back, and then dissolving it in the seawater, then adding a fraction of this enriched seawater to our incubations, and that would give us a constant enrichment over time. And then you can see here, this is the black dot here that gives constant enrichment over time. So with this, we sent a couple of grad students out to sea to test this method, and they made lots of measurements, 117 bubble addition comparison with this solution method that we designed, and we're able to show that there's indeed like a real underestimation of these rates. So we measure much higher rates. So the community that we were looking at could be divided in two groups, one that was trichodesmium dominated and one where we had mainly other groups of diazotrophs. This is a comparison of the rate, cross-section of the rates. Here you have the bubble addition method, and here the dissolution method. You can see most of the rates are higher in the dissolution method, and especially in this region here, the lower region, where we found sometimes that we could detect rates where we couldn't with the bubble method. Overall, all the rate measurements, if we look at this in between the various regions, in the tropical, in the equatorial region, we found a much bigger increase, about 5 times on average between the two methods, than in the tropical area where trichodesmium dominated. With these measurements, we were able to make some prediction about the budget in the Atlantic, which we measured from the bubble method or with the dissolution method, so we were able to increase this quite a bit. And then assuming that this could be then extrapolated to the global ocean, we said we could increase the nitrogen budget to the contribution of nitrogen fixation to 177 teragrams of nitrogen per year. But in a sense, this is just a number. It's not this one aspect of why this is important to get a good measurement, a rate measurement method. But what's more important is that we need to be able to measure nitrogen fixation so that we can see how it's going to be affected in the future with global change. So when we have ocean acidification or more dust deposited from the atmosphere and so on. Can the historical data be corrected? This is something that was asked very frequently by other investigators. So what we think right now is that we can take the data as a lower limit, but there's no easy conversion factor to correct for the historical data. And I'm going to use the 1 minute of my overtime here to show you why this is the case. First of all, the incubation conditions will vary. So field conditions vary. So here you have a nice calm day. Here you have a very stormy day. So your bottle is shaken differently over the 24 hours. So the equilibration time will vary a little bit during that time. But more importantly, there's a community composition effect on this underestimation problem. Is that if you look at the equilibration curve here of the isotope in the water, and then let's say you start your incubation in the morning at dawn, and you have a community that's dominated by trichodesmium, which fixes at midday as a big nitrogen fixation peak, then you make your calculations with this IN2, the theoretical value, and then you only have this type of enrichment in your bottle, then you're going to have a large underestimation. In contrast, if you start your incubation in the morning again, but you have a diazotroph that fixes in the middle of the night, 12 or 15 hours later, then you're much closer to the theoretical value of your enrichment, and then therefore you have a smaller underestimation. But either way, you'll be underestimating. By publishing this kind of paper, you don't make a lot of friends, but we try to compensate we thought it was very important to put these results out, and then in order to compensate for this and bring in the rest of the community to solve this problem, we held an international workshop last February in Kiel, and it was jointly sponsored by SOLAS and NSF. We had participants from Europe and North America, and we found out already there that some of the other groups had corroborated our results. We discussed developing a common protocol and then doing intercalibration crews, and then we applied for working groups for continuing this work. So I think that we will be able to come up with a new method that will work, that we can sort out the problems before we start applying it widely. But this might take a couple of years to get to this point. So what I think some of the priority areas should be, in the future, well, the method is obviously something that we want to do. All these isotopes that are only characterized by NIF genes, and we need to isolate those organisms and then try to figure out what their physiology is, what other things they can do besides nitrogen fixation. This is really important for trying to figure out how they will be affected by global change in the future. Then also, this measurement of the NIF problems here, like what are we measuring exactly? Well, we need to start standardizing these methods, DNA extractions, etc., so that we can go work as a group and then cover the whole globe. If you want to look at global processes, then you need to go sampling globally, and then there's no single group that's able to do this in the scientific community. So with this, I will acknowledge the people who were instrumental in carrying out this work. And this is Victor Moore here, who discovered the underestimation. And most of the people here have been involved in measuring NIF genes in various parts of the world. Toby Groskop is the student who was rushed out to sea to try to do this comparison of the two methods in a very short time, and he did a very good job with this. So that's all I have to say for today. Thank you.
Thank you, Dr. LaRoche, for excellent presentation. We have time for question.
Great talk. Can you comment on the acetylene method? Some people did that. They are trustworthy or even worse.
Okay, so the acetylene method, well, it's not used very much in the ocean except for measuring I think historically for measuring trichodesmium, because there you can concentrate the trichodesmium into a smaller volume and then apply the acetylene method. But it turns out it's not as sensitive as the 15N2 method. There's also a lot of problems with the acetylene reduction method. If you look at 15N2 addition, there you can have a true tracer addition because the N2 gas is very abundant dissolved in the ocean. So when you're adding 15N2 gas, you're not really disturbing the conditions, the amount of total amount of substrate that you add in your incubation. But with acetylene is like an alternate substrate, and then it could also block eventually nitrogen fixation. So some people are trying to make this method work. It's, of course, very interesting because you can get real-time measurements as opposed to the 15N2 method. So this is the balance between the two. But I think the sensitivity is what prevented the wide use in the ocean. Hope that answers the question.
Okay, other question? Okay, thank you very much for your excellent talk, and thank you very much for your attending a wonderful talk.